All right, I think this is the first time I've had a breakout session that's live streamed, so they've encouraged us to use the mic, by the way, for everything, so there are a couple that are floating around. So Ajay and I are happy to lead the first HCA breakout session for the CCF. It's great to see that this problem has particular attention for this meeting, and thanks everyone for coming here. So Ajay and I put together a few questions that we hope to cover today. There are a couple that are of particular interest to me. I'll read through them now, and then we can start on some of the most interesting ones for discussion. But we wanted to talk about, a couple of really sort of general questions that are going to apply to many of the organs and scales that we deal with. To think about sort of mapping resolution and how fine scale we're going to try to map different subsets of data. Can we construct a single CCF across ages and genders, or do we need multiple frameworks? Do we need a male CCF and a female CCF for the whole body or for individual tissues? Existing anatomical standards and frameworks that we should consider when building a CCF. How we can deal with dynamic tissues, which is a particularly fun challenge, like the heart and the lung that are changing on the microsecond level. A lot of us are, of course, dealing with molecular data, and that's what's been one of the focuses of this meeting, but I think there's an enormous role here for CT, MRI, and other virtual anatomy and physiology databases that anatomists have been building for many years that we should be considering. And one of the sort of big existential questions to me is how do we use a CCF when we only are going to sample a very small part of the organ and we have very sparse coverage of what we're trying to cover. So these are some of the key questions. The one that I've left out, which is what unifies all of this, and what I hope drives a lot of our discussion, is why do we need a CCF? What are the biological use cases? What are the questions that we're going to try to answer with this framework? And if we have a good sense of that, that can help guide our discussion. And I will say that those have been, it's not so easy actually to answer that question. Why do we need a CCF and what do we hope to use it for? And it's something that in HubMap we've been trying to define. We know that we need one, if for nothing else, to have a user interface where people can click and see where data is coming from. But what biologically do we need it for? And so I'd really like, especially if they're biologists who are interested in using such a reference here, I'd love for people to weigh in on what you might use a CCF for. And if we can think of a few use cases, then we can start to ask more methodological questions within that. Oh, actually, we have to give you the mic.
Yeah, so at the end of the day, I think comparison is something that's going to be very big for us. Comparison against, between, healthy individuals, comparisons based on genetics, and of course when we go to disease. And when we're comparing two samples, and, I just think that people haven't thought enough of what are the factors that can affect differences that we see between two samples. And just, saying, okay, this sample is different than that sample. Why? Well, maybe because it was collected from a different place. Maybe it was a different piece of the tissue in the organ. And I think that without a CCF, a comparison between individuals and saying this is the same, similar, or different than that is going to be meaningless.
Yeah, and I'm sure, you brought this up yesterday. You highlighted that a very similar problem is in the molecular space. How do we compare single-cell data sets when we have many of these same issues? How do we collapse? How do we identify shared biological properties in different ones? And this is just another covariate that goes on top of that. But I agree. That's the number one priority is to have a common reference map that we can use to compare.
We need a spline or some way of splining between different stages of pediatric development so that you can go from tissue to not just one common network, one common reference, but something that you can spline back to developmental tissues and all the way back up to adult. So that would be a different type of dynamism.
Yes, as if the problem was challenging enough for adults, but to connect across developmental stages.
To Diane's point, there's actually, we know that from literature reviews that Rahul and myself and John have done, that there's actually a lot of work on how you map over time CCFs to each other along development, and it actually connects also with some of the nice computational methods by which you align things between individuals. So it's not necessarily as distinct as it is. my sense of the developmental data that is being collected is that the temporal density is not the same as people have developed for model organisms. In a model organism, you can really have density in time. With the data that seems to be collected in the developmental atlas, at least, it's not necessarily there, and it's not surprising that it is not. In the pediatric sense, I have less sense of exactly where it's going to be just yet.
There's also a big use case for regenerative medicine. So people are very interested in developing cell therapy or repairing tissue. In general, they want to repair tissue, but you have to know which parts of the tissue are different to repair appropriately, right? So you can't just put cells from a tissue in general to anywhere. You might have to know exactly anatomically where it goes. And obviously that is more important for structures, like if you have branches or other ducts and things that you're repairing.
It's interesting how each of those cases has a very, very different scale of resolution that one would need to map, from connecting across development to comparing across individuals to regenerative medicine. So it's an interesting challenge. How about, can we think of specific, so those are wonderful sort of general challenges that would exist across organs, just to help guide discussion, particularly if there are any people who work on the brain or on the heart or on the liver or the lung. It would be helpful if we could think of a couple of specific use cases of a type of comparison that you'd like to do, or of a particular region that we would expect to be different over time. And anything to get started.
So again, going, there's differences that are defined by features such as, like, bringing an example such as the lung, as the branching, and as you sort of switch scales, I think that there could be other differences that, again, and I'm very molecular in my thinking. I'm very narrow-minded. So I like understanding why I see, the molecular details. And for example, using, not major landmarks, but distances from endothelial cells and things like that, really define the local microenvironment. So there could be, as part of the common coordinate framework, some type of, structures such as vasculature that define microenvironments that influence cellular behavior, because wherever you are, how distant you are from a vessel is going to influence a lot.
Have you seen sort of molecular, so the NIH is also really interested in the idea that there are sort of internal, very high resolution microenvironmental landmarks that govern the phenotypes of tissues, which makes sense because of course that's what's driving what they're seeing and what they're sensing. We've started to look a little bit, this is more specific, but for trying to find molecular evidence of that. So if we have cases.
At SKI has been working on, again, these are models, but he's really shown how gradients, both in cancer and in sort of more contrived artificial systems, really define huge metabolic differences that have huge impact. So gradients, particularly gradients driven by oxygen, among other things, and sort of mimicking vasculature, as well as imaging in cancer samples, have really shown that these gradients from vasculature have real molecular impact.
The other reason why that's really exciting is we can read those gradients from sequencing, so potentially if those gradients are there, we can use them to map, which is, I think, would be really exciting if one could infer that type of information.
Just give a classical example of gradient that is well known and well established in spatial and molecular. It is the one that generates the crypt-villus axis well, specifically in the small intestine, but similar gradients exist in epithelia in general. It's generated by a mutual feedback loop between stem cells and the niche. And most people usually used to think about it only coming from the niche, but the recent literature suggests it goes both ways. So that's, I think, a classical example of a molecular gradient, which is actually recoverable in work that Mor Nitzan has done together with Nikolaus Rojewski and others, has also shown that these styles of gradients are actually almost reconstructible without external information, just assuming that you know that the gradient exists. You don't know what it is, but you know that it exists. So I think those are really micro scale, and in fact, you can even think about a subcellular version of it anytime you have a polarized tissue. So for example, if you think about epithelial cells, what is at the right at one side of them and at the other side of them is actually not the same, they're polarized, and that information will be molecular and discoverable.
This might break the common coordinate framework, but structural birth defects, there are a lot of common ones, and they do differ from individual to individual, but being able to use those changes against the normal would be very useful. Like teratology of Fallot tends to be the same kind of structures in the heart.
So some of the things we almost never think about are mechanical stresses and issues like that in organs, which would differ in different parts of the organ that might have molecular consequences and vice versa.
To Ajay's point, just to say that, to my understanding, there's actually very nice new methods now to measure those that are compatible with the types of downstream molecular profiling. So you can think about the mechanical stress in a cell and then get its profile as well, which is very cool. And there are several labs I know of, including labs at MIT, for example, that have pursued these types of approaches. Not so much for human tissue, but they're not based on an engineered system that you can only do in a model organism. So I would expect us to see more of this. And I think mechanical stresses are going to be a very important thing. Again, at the histological level, that works really, really well, these kinds of questions. And the difficulty is when you actually go up.
I guess like one of the things I've been thinking about is just sort of putting in the pitch for thinking really hard about how you could, on the microscopic level, like what sort of coordinate framework could be derived to essentially position different cell types within known multicellular structures of interests. You know, I mean, for instance, it's kind of epithelial glands, you know, versus like, you know, lymphatic vessels, vasculature, you know, in the GI tract you have like a concentric ring of smooth muscle. I mean, you know, so there's all these like very well characterized compartments that, you know, that we know that there's a normative distribution of certain cell types relative to the structure, right? And we also have a very good idea about how there's perturbed in disease states. Like for instance, if you're looking at, you know, colon polyps, and if you had a, you know, a CCF for defining like a normal gland, then if you mapped on top of that somehow, you know, a colon polyp, you would see that there would be way more nucleated cells towards the top of the gland than at the bottom, and you know, and you would be able to look to see how like the phenotype of all those things are perturbed. I don't know. I'm just wondering that, like, you know, and all this stuff's really interesting, but it's sort of like there's this whole, you know, sort of very large body of work that, you know, has sort of vetted out sort of reference multicellular structures that we kind of know are, you know, that sort of situate, you know, different cell phenotypes relative to in some sort of symmetry.
There's I thought I'd give a couple of examples of use cases that we are interested in at the sort of opposite ends of the spectrum. On the micrometer scale, one of the issues that we're interested in is pancreatic islets of Langerhans. And we're interested in the difference between what goes on in the core versus on the periphery. And this we would need to have a resolution of, let's say, 100 micrometers for that. At the other end, we're working with others on creating a heart cell atlas that you may have seen. And at the moment, we're sort of fairly crudely sampling the right atrium, the left atrium, ventricle, and so on. But actually, it's very clear that biologically and pathologically, it really makes a big difference where exactly you are within the ventricle, for example, in relation to the watershed between the blood supply and whether you're sort of at the terminal end of where the vessels are. So actually, for something like the heart, which obviously mechanical properties are important, it really would be potentially make a difference whether we have a mechanism for determining the precise coordinates of where these samples are coming from. And at the moment, we're sort of assuming that the left ventricle is the left ventricle, but of course it's a large area. We would need to find a way of precisely knowing that there's the bit that we sample is the same as the bit that somebody else samples.
I just wanted to ask, so, biology is a generative system, so it generates all these structures. Some of the structures, many of the structures are defined perfectly. Everybody has five fingers and 5 toes. But I wonder how many structures may need to be defined generatively, like a field of something with this density, or a branching structure that doesn't have to be exactly the same between individuals, but it needs to have a particular parameters associated with it for it to function. So have people looked at that problem?
That general idea of understanding the foundational organizational principles of human tissues, which is I think what a lot of people are saying here, is incredibly exciting. I think in the lung, there's been, particularly in the mouse, there's been some really interesting work to understand sort of how conserved and as the generative process that gives rise to that branching structure and the cell type specific niches that exist in different airways. I'm sure that's true in many tissues as well. I'm not as aware of it, but.
Certainly blood vessels and things like that, yeah.
Okay, so I think, by the way, if anybody has ideas on use cases, please weigh in throughout this, because those are foundational to this. But there's sort of 3 general themes that came out there. The first is the ability to do comparisons, and that can be just across healthy individuals. There's particular excitement across individuals and disease to be able to understand the anatomical basis of that. The potential to do comparisons across developmental time points, and of course in the context of birth defects. So that ability to map in a consistent way to compare is one. Another is the ability to understand the sort of environmental influences that govern cellular behavior, whether it's the particular stresses that they're under, or the blood vessels or nutrients that are nearby, or other cell types that are nearby. And the third is the ability to understand sort of from a cellular level how tissues are structured and organized as a foundational question. And of course, how that also goes wrong in disease, but trying to understand how the body is structured at single cell resolution. So those are 3 sort of wonderful use cases to think about going through. What about this question, which I really struggle with, of how many CCFs do we need? And one that I really think about is, do we need a male and a female CCF? And more generally, sort of when we think about constructing a common coordinate framework, at what point do we start to worry that we are over-collapsing inter-individual variation that's essential in this goal of becoming common, and how do we start to make decisions about when we need more than one, or if we need more than one?
And that's a tough question, Raul, but I mean, just saying the obvious here, male and female have different organs, reproductive organs, just to start with. So there's obviously a great need for having both a male and a female. Not to mention that size of the organs are different. Composition and structure may not be different, but we don't even know that. There is a series of diseases that are more prevalent in women, such as certain type of autoimmune disease. And it'd be interesting to understand at the cellular level if some of these structures are different. So I think there is a need for both a male and a female CCF.
No easy answers to this one?
So the question I would ask is, what is an example of a CCF where someone would envision that, like a specific like paradigm for a CCF that would not map between genders? Like, because I'm just sitting here thinking, I mean, because it's kind of like, because you're talking about CCFs in terms of, like, for instance, like you showed for the lung branching structures or whatever. I mean, by the nature of the way that you're doing it, even if there is differences between, male and female, that by the nature of how you're mapping it, it's going to be captured, right? And so, I mean, I guess it's, so I'd be interested to hear an example of when someone thinks it, wouldn't map.
This isn't an answer to your question, but I'm thinking about it while I answer this, or say this. I mean, we might need different scales of CCF. So we have a scale that's specific to the heart, and then, I mean, we might have to have levels of granularity. So between there are going to be physiological body differences between men and women. But if you take the heart and you do your common coordinate framework for the heart, where it places in the body may differ between men and women. So you may have an anatomical woman model, anatomical man model, and then have the different pieces of the CCF map inside those models, which would help balance out what's needed. That's just...
That's certainly one thing that we've thought of, that you would have, I mean, because of course you're going to have different anatomical models for obvious reasons to begin with, but it may be that some of those anatomical models would then map to the same atlases. For example, if we think that the lung structure is relatively similar, then you have the ability to map general.
I suppose that that sex is one factor, but there are other factors. So maybe it could be factor-oriented. So based on variability, like you have, you know, certain translations that need to be made in different ways depending on whatever factor you're considering. That might be too difficult to genericize or whatever, but.
I think to generalize what you said, the question is how we model variance generally. And I think that's a question both for the CCF, but also for you know, cell types in HCA. We can define an endothelial cell, and it's going to have some mean expression profile, but there's going to be some level of allowable variance in there for what we consider to still be endothelial. So there are going to be key features that are going to be conserved across everything that we give a label to, and then there's going to be allowable variance. How we model that spatially is something we don't know how to do yet, or I haven't figured out concretely how to think about it in the context of the CCF. But it highlights, I think, the importance of starting with a mean representation, but then having a probabilistic mapping on top of that.
Some of these things you might have to get mathematicians involved who are interested in topology or something like that. I don't know. You know, I'm not an expert in that area, but people may have figured out some of these things from a geometric point of view or something. I'm not sure. Does anyone else know?
Can you take it? Oh.
Raul could have commented similarly. I think historically the approach was very mathematical in this sense. It was kind of model-driven rather than data-driven. So people would first make an assumption on something, right? It's shaped like this or it's done like that. And then they would try to model the organ in this way, and then they would make mathematical transformations that would shape it all in this direction. And I think there are newer approaches now that we're definitely looking into that make it more of a data-driven type of problem. So you've seen one example that Rahul illustrated yesterday where you assume that it's a fractal because it's known to be a fractal, but you don't actually work on the data directly. You train a model on simulated data that just has the general features, and you can imagine you could do that in other organs as well, not with fractals, but with other features like that. And then you actually do everything in the context of that model that you trained. Another approach that people have started taking is using optimal transport, and which can be done again on the data as it is or in a latent space where the data the image is already embedded in some way, in order to relate different things to each other just by saying here's a distribution in space into another distribution in space. And it's useful to know that when the optimal transport problem got better solved from or approximated, so you could actually handle it computationally around 2013. One of the earliest applications was in computer graphics, precisely in order to write, these things where they start with an image and it kind of morphs into like with one creature and it morphs to another creature. That's an optimal transport solution today. So it fits really well to this purpose, and people started doing it with CTs and MRIs.
I'll highlight how exciting it was to see this sort of model-based approach be successful in the context that we were looking at. That was an idea, actually, that at least I proposed, or even I think you did.
Not what we originally proposed. Ajay, I think, is not going to blame us for that. It's not in the proposal.
But actually, Ajay was excited about that approach because, of course, if you can propose a generative model, it gives you all sorts of analytical options that you have, rather than if you have sort of an unconstrained tissue that you're trying to work with. So I think that's the lung is a great test case for this, to see what we can do with an imperfect but still very highly analytically tractable model of the underlying structure. And then that gives us a natural platform for working with differences across ages, with differences across genders. And so I think that'll be very fun to see how that works. Of course, generalizing that across tissues is going to be a challenge, but many tissues can be modeled with interesting processes.
I like what you said about the embedding or the latent space, because obviously that ties to machine learning, and I don't know what people have, you could have an autoencoder or something like that that learns it based on all the data that you have. But you have to have a lot of, I guess, representation of the anatomy in some way that it can be input.
I think one question that Mike asked, which if anyone has good examples of, please weigh in, is in this context of gender or inter-individual variation, we should start to think of test cases beyond the reproductive organs, which are really sort of a discrete case in type A and type B, where we know that a framework that's going to be learned in the context of one person will not apply to the framework learned in the context of another. So for example, if there were known differences in anatomical niches across genders in the heart, which I don't think there is, but if there are cases like that, then that's something we should be keeping in mind sort of up front to think about how to model that type of diversity. It's not something I know as much about, but.
So along those lines, I guess the question I have is that if you, because I guess like just intuitively, I've just been thinking that ultimately this a CCF is going to be structured sort of as like, it's kind of Russian, nested Russian dolls. Like there's going to be, increasing layers of how you're going to have something to define organ location, and then you're going to have something to define like relative location within organ, and then you're going to have something to define, finer histologicals. And I guess the thing is that I'm wondering is that if you do that, it seems like that would make it fairly robust against a lot of these variants. And I don't know, I'd be interested to hear if people have like dissenting opinions about that, about like why it wouldn't be, in what cases it would fail.
I think I agree. It's good to start thinking about actually what variants do we want to map, because there is an interaction between the CCFs and the maps. So it may be that, let's say that there's only one CCF for the heart because there are no appreciable differences, but actually there will be a male and a female map because the cells will be different, even though the coordinates are different. So it may be that for some maps, the CCFs will be the same. So if we wanted to map, maps that represent different races, for example, which may well have different biological significance, it may be that we use the same CCF, but it may be that we need a different CCF if the variance that we're interested in is age, for example, because of natural and anatomical variations. And speaking to that, I mean, we're talking about space most of the time. We've mentioned age as a sort of, I guess, a map of time. But it is also worth thinking about how detailed we want to be able to map these variances. So taking time, for example, well, age is an obvious example. But there are very important differences in cells and tissues across the circadian rhythm, the 24-hour cycles, right? Do we need ultimately a morning map and an evening map because the cells will be different? Do we need to have maps for different seasons? Is our biology different in winter compared to the summer? And then we have to be pragmatic. It's impossible to map all of these at the same time, but at least it's useful to start thinking about what variances we should take into account and which ones are then on our priority list.
Some cells should have like a fixed, stable positions, right? And so that should not vary across season and time. But other circulating cells, such as immune cells, will definitely be all over the place, depending on when you take your snapshot, especially if they're circulating cells. And so from that angle, this could almost be endless. So maybe that should not maybe be the initial focus for the CCF, although like would be tremendously useful. Speaking from someone who's part of the Immune Cell Atlas, would definitely be very useful, but not maybe very feasible initially. I do have a question to Raul and to other people quite involved with this. How do you think about the structure, these organs that are very structured to start with, that have clear landmarks and repetitive elements, such as the liver and the kidney, which perhaps are a good use case to start because of that structure compared to other organs where the repetitive elements are not as clear? How are you thinking and approaching this in terms of prioritizing efforts?
Certainly from a practical perspective, we're going to start where we have a better sense of how to begin. But I think, Kareb said, am I pronouncing that right? Chris. It's the question of what level of variation do we want to map. And that's a biological decision that we make for what we want to focus on. And the level of variation that we decide to map then tells us what types of comparisons we can do downstream. The finer scale variation we type the map, the more fine the comparison that we can do, assuming that makes sense. Just before getting to that question about unstructured tissues, actually one thing that's been actually quite helpful here is this idea that we need to, I mean, we've always thought about having a hierarchical CCF structure, but I had always thought of it more as a tree-based type system, which it may be, but what you're kind of saying is, every person may have their own map, and every group may come up with their own map of their own individual sample, and what we need to build as a community is some way for them to insert that map into a broader framework. So if someone wants to build a map in, December versus March, they can build them separately if they want to, but then they should have the ability to plug that into something that's more conserved, and then you can see whether things change in December or in March. And that is also very similar, again, to this problem of, I like trying to draw as many analogies as possible between trying to draw single-cell reference maps of different types and states as a CCF, where you would sequence your own sample, but then you have the ability to classify your cells based on references that other people have put together, and those references can be T cells, B cells, or CD4 T cells, or CD8 T cells, and get finer and finer and finer. So I think that's a good analogy to have, this sort of hierarchical classification structure. I'm open to any and all suggestions on how to deal with unstructured tissues. Particularly things like obviously anything where cells are traveling at a high rate. This becomes particularly interesting. So, outside of saying that we will always start by looking for what is common, and if there is nothing that's common, then what is the value of constructing a common coordinate framework if you can't do any comparisons if nothing is common anyway. That's as far as I've certainly gotten in that context.
If I may just go back to two related points. I think your point about practicality is really important. Clearly, we don't have capacity to start creating March versus December maps at this stage. But I think it's important for us to record that data, so that actually in 5 years' time and 10 years' time, somebody can go back and say, actually, I now want to look at all the data that was gathered in March so that I can create my own map. And it would be a great shame if we get to 10 years' time and suddenly realize that actually nobody's been recording time of day when the samples were processed, or time of season. So even though we are not actively creating those maps, actually capturing the data at this stage makes it future-proof, I think.
I was just wondering, like, if we're kind of sort of mixing together like the idea of like unique maps for different scenarios versus the CCF framework, right? Which is that like for instance, even with immune cells, which are constantly transient and all those other kinds of things. If you look at primary lymphoid organs, there's very fixed, consistent multicellular structures. There's germinal centers of the interfollicular region, which will always be there, right? And so the question then is that like if you look at it hierarchically, it's like if you look within germinal centers across states, then which cells are present within the germinal center. But the whole thing though, that they're mapped to like fixed multicellular structures, right, in every case. And so I guess like to me I think that like there's a difference between taking into account all these different variations when the maps are being done versus how those features are mapped onto a common coordinate system, which could apply to all of them, even though the players within the reference structures may vary.
I think where this gets particularly important, and it would have been nice if Vee was in the room. One is the collection of phenotypic metadata, and the good news is, as Laura points out, is that's something that in any data set, regardless of whether it's spatial or not, you want to collect as many covariates as possible so you can go back and check for that. But one of the particular challenges with the CCF, especially when we're dealing with the fact that there are multiple labs and multiple consortia involved, is how to harmonize sampling strategies and anatomical region dissection and that sort of thing as early as humanly possible, because once you make that decision, you can't go back and change it. You can always go back and check, record the time of sample collection, but you can't retake it from a different location so it's consistent with something else. And assuming we aren't able to come up with some sort of magical solution that will take a single cell profile and map it back to its precise location in the body, we will be highly dependent on the spatial metadata that is collected and the sampling strategy that is agreed upon relatively early. So if people have ideas on that, especially coming from a tissue collection process, that's great. Our general thought is it's important in the lung to get as early as possible the right community of people across different projects together in the room to think about what can be unified in the context of sampling.
To say a couple of things. One, respond to Karosha's earlier point about different days of the week or whatever. I guess from a machine learning perspective, you could, and the data-driven perspective that Aviv mentioned, you could learn all of this, and you would have an encoder and a decoder, and it would learn the factors that are actually varying, and you can map to a common space. That requires probably a lot of data. You might not have all of that data, but you could do it without knowing even the factors and then interpret them afterwards. So that's well worked out in machine learning. But on the other hand, you have the model-driven version, which is you know what the heart looks like because we already have anatomical maps. That's much easier for us to work with for databases, for instance. It's not easy to capture internal representations of deep learning systems and understand what they mean. So that's probably a research topic. So from a general perspective, you might need that in the future to capture all these variants, but from a practical perspective, you need to start with something that's interpretable and easy to use and easy to encode. And so I guess that's a factor in how to start. The second point is in answer to your question about what to do from a sample prep. We had some discussion recently in the liver project. There's a group in Austria that is looking at, basically provided their feedback that they would like to see everybody take 3D images, medical image type things of the organ that they're taking the sample from, so that it's just recorded in a somewhat automated way. I don't know exactly how to implement that, but they do, and they're already thinking about it for their project. So that's interesting.
So that would be transformative for mapping, right? If there was an agreement as much as possible to try to take, CT or MRI, and in the context of the HubMap lung, that is something that they are doing. They are starting with 3D reconstruction CT, and then doing the slicing so we have image data to go back and see where data was collected from. That's not necessarily in either the budget or the purview of every single lab, but that's one of the reasons we chose to start there, because it gives us always a reference point to go back to, and I hope it can be done fast.
Speaking to that point, getting CT, for example, imaging would be great, but in many cases, this may not be possible if you don't have access to the whole organ and you simply you're sampling it in situ. So one of the things that we are planning to do, it's not straightforward, is to do a video imaging at the time of sampling, so that actually the person who is taking, there's real-time video, so that actually then that video we can go back and interrogate it and try and then retrospectively assign that particular location to a coordinate that is created at some point in the future. We've thought about still imaging, but we feel that actually ultimately video imaging would be great. So the surgeon who's actually taking the sample, for example, might be wearing a head cam, or there may be a separate person who's actually videoing it, so we can go back and.
So on the same point, I mean, one of the things that I heard one of our surgeons complain is that all these CT images and lung images are for inflated tissue in the body; they're whole, and the moment he places it on the slab to cut a slice, its shape is entirely different, and you can no longer necessarily look at the CT image and look at the heart on the and know where is what.
surgical group that I'm working with who's looking at kidney cancer is trying to solve that with 3D printed heart structures that they can put the samples in standard ways. I don't know if they've fully figured it out, but they are sampling based on putting it in kind of this 3D printed plastic structure that it can be controlled at least. I don't know if anyone else is thinking about that.
Can I ask, is Kristen is here? In GTEx, do you guys, is Kristen Ardley here? Or anyone from GTEx? Okay, I'm curious how they handled this particular problem. If they tried to collect spatial metadata when they take samples from tissues, and it's something we should find out more about because they've thought about a lot of these challenges in taking data.
So I'm not part of GTEx, but we do rapid autopsy where we collect full organs within 30 minutes of passing. And in that context, we actually take pictures and record the precise location of where we collect samples. With more funding, we can actually do more sampling. But to go back to your point, Raul, about sampling, I really appreciated what you presented from the lung group, where you were where the group is so systematic about mapping all the location and dividing it per sections. I think it's a great framework to be expanded to their effort, and it's pretty clever what the group is doing with you for kidney cancer, for the 3D imaging. I think that's really thinking outside the box. That can be a good solution. Just talking about circulating cells, Raul, you were asking about questions about how you do about circulation. I don't know that's a perfect solution, but what we started doing is teaming up with pathologists and radiologists, and we use PET scan imaging, tracking secreted factors related to immune cell, like granzyme B and perforin, and to try to see if their patterns emerge within organs. And it's very early, but it does seem to be emerging, and we track it with some of the live biopsies we're collecting. So they might, I mean, this is too early, but there might be ways of thinking about circulating cell. And just to get back to one of your earlier question, I actually completely agree with your hierarchical approach to thinking about these maps. And to point back to Michael, I mean, honestly, about the immune system, you're right. Like germinal centers, there are parts of the organs that are very structured, right? It goes back to how do you prioritize which ones to look at. So anything that's very structured for the immune system would work. Anything that's circulating would, I guess you would need several maps over time. So if the group is going to come up with frameworks to empower the broader community to engage and build on it, that would be tremendously useful if we had some guidance from that aspect so we can all pitch in.
And of course, there's a need for practical solutions to this problem of how do you record when you can't take a picture. One of the ideas, and actually this wasn't my idea, it's an idea from my sort of mapping center co-lead, Katri Borner at Indiana, is to provide surgeons with high-resolution 3D models of tissues that have been built by anatomists and tell them to point to where they took the tissue from. And it's not a machine learning mapping strategy, but as a starting point in terms of something that at least is highly interpretable, we trust the sample collection procedure to know where they took from. And at least that gives us something to go back to. And so that's actually not the worst starting point to begin.
Has anybody thought about the sort of gold standard experiment that is GTEx-like, learning from GTEx and trying to do sort of, for even a small number of individuals, you have to be careful to be sort of equitable across ethnic groups, sort of whole body autopsy, very precise information, maybe not quite so high quality in terms of RNA because you'd have to take slightly longer over it. So you have some ultra precise information for a small number of individuals. You know, on a sort of longer time frame, just while we're trying to make best efforts in the normal run-of-the-mill experiments.
I think the more that we can think of sort of the ideal pilot test cases, the better. We picked something that, in the context of HubMap, that we thought would be a good match for some of the methods that we wanted to build. But if there are people who are designing experiments, particularly with the spatial context, and can think about how to maximize the value you could get out of that type of experimental design, please get in touch, and it would be a wonderful place to start. Are you raising your hand? Okay.
So on that note, we look at distal lung structure a lot, and I think I'm sort of wondering from your guys' perspective, so you have the MRI or the CT, and that only makes it so far down, right? And then there's there can be heterogeneity in the distal lung, there might not be, depending on the situation, and it changes over time. And so sort of once you get down to that direction, is it all distal in the CCF that you're thinking about, and sort of that's the unit that's going to be down there, or sort of for these things where it's going to be difficult to connect the MRI or the CT data to where it came from? Have you thought about how you're going to integrate that data at this point?
So I think that, to be completely honest, the best idea we have, if you aren't, for example, able to place a sample that's taken in the context of an existing CT image, then the best idea that we have well, there are two possibilities. One is someone tells us where they took it from, and the other is we figure it out. And to the extent to which we're going to have the ability, based on whatever the sample type is, whether it's a spatial transcriptomics data set or a single cell RNA-seq data set, the resolution that we're going to be able to map back to is probably not going to be incredibly high in the lung. In the brain, I think we'd have a lot, we have layers and there's markers of anatomical structure. In the lung, we're less certain. So, but that's something we're gonna have to find out. I think another thing which we haven't talked about really at all is the context of and the concept of ontologies and how those build into sort of categorization of a CCF rather than necessarily a numerical sample for where things come from, and how much we should be thinking about, rather than saying this came from coordinate X47, Y56, just say this came from the end of the right bronchus, just something where that may be the actual primary anatomical consideration that most people care about. And then the question is, how do we come up with the consensus set of those terms, of those categories, which would be much easier also from a sample collection standpoint to define, right? So I took it from this because this is something that we've agreed upon as a field. So that's also in the context of HubMap. We have two groups that are involved in the CCF construction, and one is particularly focused in the context of ontologies. But that seems like a solvable and potentially extremely high resolution, particularly for the types of comparisons that we want to do, because then you could compare, for example, all data sets within a single ontological category. So that, again, is a sort of tissue-based community effort to define what your set of those ontologies would be. But those are things that people have been doing anyway and are doing already in the context of many genomics projects. So I think that's a nice option.
Sorry to go back to the previous topic, but I have a comment on the live chat from Anna Hardina, and she said that, yes, we are working how to record exact sampling position in the liver, and we are testing workflows to get imaging and sample position information on multi-scale levels.
Oh, wonderful. Okay, so we have a nice. So I think if people are working systematically on that problem and are also generating the types of single cell data that you're working with, then whether it's through Slack or whether it's through email, we should stay in close contact as using that as a pilot opportunity to see how this works. The best, I think, eventually, one day, we will be able to learn these things from the data itself, but the more ground truth we have, obviously, the better we're going to be able to train those models.
I just wanted to make a comment to your ontological annotation as well, that it also gives a really good backup to a coordinate system. I mean, I worked previously in the biodiversity field and spatial coordinate systems like at landscape levels. So often when you would look at the spatial coordinate and it didn't back up actually ontologically what was written in the same field in the database. So you might, and in particular, in the biodiversity field, one of the most biologically diverse areas on the planet is at 0, 0 in the middle of the Atlantic, because that's a coordinate that often gets assigned by default. So you need both, I think, to be able to do it accurately.
Yeah, and I think the way that would work would be to define a series of ontologies, categories, and then in the context of visualization and anatomical comparison, you can assign them to a coordinate point in the human body, but what you really care about is making comparisons within that grouping. And that's definitely, not only a backup, but one of the strategies we'll pursue heavily.
I know Danny and Simon, who are the ontologists in the HCA DCP team, are looking at this. And I think Uberon as an ontology is fairly anatomically driven, but there are a couple of other ontologies that are slightly more surgically focused and there's sort of slightly different levels of detail, and they're looking at the best ways to sort of bring them together, and if across all of the different atlasing efforts, we can build on the established ontologies that are already there and make sure we have a sort of grouping that makes sense for what needs to be done for atlasing and gives the surgeons the right tools to pick things. So I think for HCA, we ask people to give us as precise an ontology as they can, and we do the term mapping for them at the moment. But and so some places we get super precise, other places where it's the liver.
So can you say any more about two things? One is how you harmonize or how you think about that process of harmonizing ontologies across different consortia, and the other is how you do the term mapping.
So the term mapping, this is where it's unfortunate we don't have any of our ontologists in the room. The term mapping is being done by a combination of Danny and the Wranglers, although the EBI has a tool called Zoomer, which essentially is a set that can be trained to use text strings to map to particular ontology terms, and so we could create a curation across all of the atlasing efforts that was these terms mean this ontology term, and you can with Zoomer you can even specifically say I only want to trust these people's mappings, and it will give you back the right terms to help automate it, and that can even be accessed programmatically. As far as how they're assembling the ontologies go, I am not an ontologist, and so I don't know the precise details of what their approaches are there, but I can definitely make sure they're in the loop for these conversations as they happen sort of virtually.
So this is one of the things that HubMap is considering in detail in two particular contexts. So Kati Borner is thinking about this a lot in the kidney, and I think this is almost a political or a human challenge rather than an analytical challenge, right, of coming to agreement across different groups of people. And so in this case, there's a kidney precision medicine project, rebuilding a kidney, HubMap, and HCA, and then for the lung, there's Lung Map, there's the HCA Lung Consortium, and HubMap. So I think that is something that, whether it's through workshops, whether it's through Slack, whether it's through communication, agreeing on those things should be a achievable process.
Obviously, from both HCA perspective and actually from a EBI perspective, we provide ontological services to the whole community, and so being part of this effort and making sure that we can support everybody doing atlasing, I think is something that's really valuable, and we'd be very happy to be involved in.
That's good, Tyler.
So we say CCF, F is for framework, and we've dropped the word framework quite often. I don't know that we've had an in-depth discussion of what framework actually means. I mean, what's the framework so people are expecting Rahul to be building some framework. What do you want in the framework? Is that a reasonable question to ask, or is it too early?
I'm not sure what are the options? What are I'm not quite sure.
So you want other people's common coordinates to be merged into some reference common coordinate. So I presume that's one meaning of the word framework. I don't actually know what else there could be.
Well, I've got sort of GPS coordinates in my mind, the equivalent of GPS coordinates, so that I know that this sample that was taken is from this precise location, and that actually the cells that I've got either map onto that GPS coordinate or not, or the equivalent of GPS coordinates.
Yeah, so I think that, but you're right. So your point to generalize what you're saying is, if we were to build an incredible coordinate system but then didn't have an accompanying strategy to map new data back on top of that, in the end, it wouldn't be particularly helpful to the things that we need. So the framework side of it comes with the ability to take new data and place it into that coordinate system, and whether that's a supervised or unsupervised approach, that's a key part of what we're doing. I'm not sure if there's a whole lot beyond that to say. How about, let me ask one broader question too. So what are the types of data, almost everything in this meeting is talked about, single cell technologies, and with the exception of the CCF talk, where I talked about it only a little bit at the end, what are the types of data that we need to be able to generate these types of scaffolds and these types of coordinate systems? Do we have those already? Or are there new technologies and data sets that we should be thinking about generating? So one example that we've thought a lot, at least a little bit about in the context of HubMap is, do we need whole body, do we need to generate whole body MRI and CT databases to start to build these anatomical scaffolds? Are there new approaches that we need for whole body imaging that we can use in this process, or are there already reference databases that are out there? So people can talk about anything, but if people have ideas there, that's another question to throw out.
It can get expensive. I know some people who are, the mouse phenotyping projects tried to do imaging, and they had a lot of, they built MRIs that you can put 16 mice into, and they had a lot of work on normalizing, for instance, the shape of the liver depending on whether the mice was in and like which, how they were lying in the MRI. So first of all, it's complicated because of those issues. Secondly, it generates a huge amount of information that you have to process. And but there are people like the mouse phenotyping community where they phenotype every knockout mouse with a range of things, including imaging, and they map everything, and they're looking for size changes and other things like that. That people have worked in that area. So you could imagine a similar thing for organ samples, put 20 of them into a little MRI system.
And just building on this beyond the mouse, I mean, there's a whole medical community that does imaging. So are you, I mean, that's a question, I don't know, are you already involving radiologists beyond surgeons that are doing this day in, day out, that could provide you all these images for every patient you're sampling? Is that part of your plan?
So for the lung, absolutely, and they're part of the workshops that we're having and the strategies that we haven't thought about it as much in the whole body context. I think Ajay has probably thought about this more than I have, especially as he's evaluating sort of new technologies to bring into the. Actually, I'm curious, Ajay, if you have a thought on this, sort of what major technologies are needed. But but anyway, nothing you can share publicly. But trying to think about the right people to bring into the room for these discussions is valuable, and in general, I think our community doesn't talk as much to radiology and CT, and this is an opportunity where that has to happen more, and it's great that encourages it. But I think that's a great point to highlight. And
feels like stating the obvious, so you're probably already doing this, but there's centuries of work of pathology have been repertoriated at different levels of granularity, all these structures, right? This legacy knowledge. Like pathologists have great depth of knowledge about all these structures. So how are you integrating all that legacy knowledge and expert pathologists for all of these organs in your process?
So I think, as we already had, with these sort of and the lunch and learns yesterday, these sort of per organ meetings, right? And we talk about that sort of challenge of bringing communities together with diverse expertise. And I think this is a particularly hard one because it's not molecular expertise. It's not the people that are traditionally associated with our community. The way that we've thought about it for HubMap, and actually we had a lot of interesting discussions. We knew we wanted to have workshops, and we were thinking about how to structure them. And what we decided to do was, instead of having sort of a general CCF workshop where we would talk about general challenges of building CCFs, would be to have two more focused workshops, one on the kidney and one on the lung, and at least in our case, with the particular goal of being able to discuss these organ-specific challenges with people that have very diverse expertise. Many of the people that I reached out to invite, for example, I had never met or had not published in journals that I typically read, but we felt had really important expertise to contribute. The academic radiology of literature is extraordinarily extensive. So I think that's, for us, going to be a new experience. And a really, really valuable source of knowledge. But outside of sort of these in-person meetings, which are not easy, they're logistically challenging, but they're necessary in these cases, I think that's how we're getting started. I would be very grateful for other people's advice on cases where you're coming from a more molecular perspective and have found solutions with integrating into a broader anatomy pathology community that's worked well. Mike, if you
We have another comment from Anna Herdina on the live chat. She says that for the liver, they have anatomists and a radiologist working on the project.
Sounds like you guys are well set up.
Well, they're new, and we should maybe talk to them because they're just starting to integrate.
And I think then in this also these questions of use cases and these questions of ontologies, I mean, those are the communities that should drive this. And actually the other, we haven't talked about it as much, I mean, one of the things I always found very compelling was the idea that the atlases that have been constructed, for example, for stereotaxic surgery, have often been in the context of a problem where, you know, someone is looking at something on a CT scan and they want to tell a surgeon this is the place where you need to operate. So that's a very clear use case where you need to have a coordinate system and a framework to be able to go in and uniquely mark a location in the body. It's not for some genomics comparison, it's specifically to mark a region. So I think that's you know, actually, when I travel, I try to ask people, you know, what are sort of the analogous surgical procedures where you would want to demarcate something, and how would you tell a surgeon where to look? Because whatever language you would use says something about the ontologies that are common in your organ and the landmarks that are common in your organ. And so that's, I think people are obviously thinking about that all the time because spatial matters when you're trying to take a piece of tissue. So.
Just an example, though it does get a bit invasive, but in the context of different type of tumors, such as breast cancer, you do imaging and then you can insert radioisotope labeled beads around the tumors, right? So actually you define precisely the location physically first. And then when the surgeon operates, you actually have the imaging and the actual physical location. And that's the case for many different type of tumors. So that's a practical example of how you go about it physically. It's quite commonly used, actually.
So I can give an example that is actually we're moving from invasive to being less invasive in terms of surgery for the gut in Crohn's disease and ulcerative colitis. Usually a big chunk of the gut is cut out during resections and surgery. From the Helmsley Trust, we invested in 3D models to really create the structure of the gut and help surgeons operate locally, just removing that piece that is inflamed and affected. And it's been extremely helpful. At least there are a couple of patients who benefited from that. And that will create sort of a framework moving forward and hopefully helpful for the single cell and human cell atlas community.
So can you talk more about that? So you mentioned that you invested in the generation of these models. So I'm curious how they were generated, and then also how surgeons would actually use them in practice. Is it again just sort of physically marking something? How does that work?
I mean, it requires lots of work and collaborations between radiologists, surgeons, and IBD specialists to really localize where in the gut is sort of the inflammation. And what they do, actually, they print in 3D models, the gut, and relocalize it. And they train robots to help them do the surgeries. So it's robotic surgery instead of being just classical GI surgery. Of course, it's not based on the molecular aspect, it's more on the imaging. So they take 2D images like CT, MRI, as well as endoscopy images from video capsules, and actually they build and scale it up to 3D. So this is what we're doing. It's mainly mathematical and machine learning at this point.
Yeah, I love that idea of having sort of 3D printed sort of physical a guide that people can use. How they build those models, I'd be very interested to learn more about that, because again, you have to build a model that is effectively capturing the common features that are going to be present across individuals. So that's where it gets really interesting.
People in companies like Autodesk who do CAD and software and they build buildings, they do these things, and they actually have they have people who are modeling the human body that are useful. They're using that for computer animation, like movies and things like that, where they want to animate people or whatever. They might use it for medical applications. But there actually are people who have computer graphics skills from computer science who are working in these companies designing systems, and they'd probably be interested in chatting about how they do those things, because they've worked on it for a long time. I've seen some of that work.
So I'll throw out just we have another 10 minutes or so, a couple of the questions we didn't really get to, which are very challenging. The first is, we didn't really talk a lot about what to do in the case of dynamic tissues. I mean, you mentioned it was challenging for surgeons who would take an inflated tissue and then it would collapse when you were trying to take sample. And how should we think about that? Does it even make sense for us to have a specific coordinate system for something that's changing every millionth of a second? And the other is how to deal with the fact that we will be collecting a very sparse part of the human body. We will not be sampling this at high resolution. We will be sampling. It's very unlikely that we would take the same anatomical location from two people, really the same homologous location. And so does it even make sense for us to map to the level of the, to micron scale resolution when we're not going to be able to do an anatomical comparison at the micron scale resolution? That obviously is going to change as technologies generate larger and larger fields of view, but it's a challenge for us to think about right now in terms of the types of frameworks that we want to build. So I don't have answers to either of those two questions, but would be curious what people think. Those are hard. I think that's also a case where sort of the scales that are generated across the fields of view that are generated by different technologies changes so much. So obviously, whole body CT and MRI, then you would be able to do comparisons across the whole body because that field of view is there. Not necessarily at the molecular level for single cell or spatial technologies, but so trying to bridge across those two scales is something I think we don't know how to do yet. Anything else you think we should cover or try to highlight or summarize?
I think it's clear that there are lots of groups who are doing very high quality work that relates to this. I wonder whether it's useful to have some sort of a central database or where people can learn from each other's experiences. We've just been hearing about this good work that this other group is doing. And I think there's a sort of danger of everybody reinventing the wheel. It's a matter of sort of sharing that expertise, and if somehow that could be coordinated, that would be useful.
So I'm realizing in retrospect, even for this session, it would have been useful to have a CCF Slack channel. A Slack channel is not going to solve this problem, but at least it's a place where people in this community, if you have ideas, particularly online, if you have resources or literature or ideas that you'd like to contribute, at least it can be a clearinghouse for that. Certainly when we host workshops, we will advertise those there, and those will be open to the community through live stream if people want to join and get involved. I think this will start to become more concrete when we start to release pilot frameworks. So the second that we release a UI or a pilot CCF for a tissue, I'm certain then that the broader community will get involved because they'll want to map their data onto that framework, and they'll have particular expertise or things that they think are missing. The question is how to get as much expertise in the room as possible before doing that, so that it's as useful as possible to the community. I'm happy to end there. Ajay, did you have anything to add on top of that?
No, I mean, I was going to raise the same thing about what we would do tomorrow when we go back, right? It's the same question that you generated. And I'm not sure. I mean, even the HCA Slack channels are mostly quiet. I mean, it's not, I don't think it's so. Maybe some idea like a central place where, so you guys have been doing this literature review and things like that. Maybe we should start by advertising that in some manner and ask people to contribute. Maybe a wiki might be a more, at least for a while, these things last and then they all sort of peter out, but I don't know.
I'm aware that this kind of risks creating a lot of work for everybody, but I mean, is there a need for an equivalent of at least something like a white paper on CCF where you sort of summarizes strategy and some goals and some priorities and identify some people who are active in this area and perhaps some contacts where people can reach out?
So I think one of the sort of key takeaways from all of this is the way to get people involved is you have to produce something, whether it's a wiki or a literature review, or whether it's a pilot CCF, or whether it's a white paper, and then give people the opportunity to contribute. Trying to ask for contributions in sort of a white, sort of open Slack channel is unlikely to yield a lot of engagement, but if there's something that people can comment on or add to, then that really helps. So there are a few sort of general resources that are already out there. The first is, which we refer back to quite a bit, it's not really a white paper, but it's sort of a summary that came out of the NIH CZI HCA workshop at the end of 2017, in Bethesda. And it's actually nice to see how sort of intellectually things have proceeded since then, but that covers sort of a lot of the key questions that we talked about today. From the workshops that both Katty's group does in the kidney and that our group will do in the lung, we will also produce sort of summaries and write-ups. I don't know if we want to have a specific CCF white paper, or actually if we even want to have a specific CCF working group, because we don't actually have one for HCA, although my talk yesterday was in the working group update. So that's something we could consider. And if we felt that that was something that would attract broad interest across the consortium, it's something we could certainly consider setting up. I don't know how that process works or how Aviv would feel about that, but that could be sort of a unifying piece of infrastructure to bring this type of thing together.
Just speaking to that and the updates we had from the other working groups, for example, on the ethical side, I thought that was actually fantastic, and that really seemed to me, as a sort of looking at that, is that actually they were making great progress by having those working groups and having a sort of a clear strategy. So I think I would be supportive of a CCF working group.
So let's do this. Let's start with the Slack channel, because at least then there's something there. So I will make that actually now, between now and the breakout session reporting. If people are interested in staying involved, potentially being a member of the working group, potentially thinking about contributing to a paper, just feel free to write a comment in that channel, so at least you have a way of sort of registering interest. And at least there's some forum that we can use to communicate on this issue. We'll think about the best way to keep people engaged and to bring sort of communities together, but at least that's a starting point. I think that would be useful. Okay, happy to end there. Thanks very much, everyone.